Certainly. Here is the text you provided. Primary nutrients and energy production. Overview of energy sources. Cells primarily utilize three macronutrients for energy: proteins, fats, and carbohydrates. Each plays a distinct role in metabolic pathways. Carbohydrates are often the first source of energy, broken down into glucose, which is then converted into ATP through glycolysis and the citric acid cycle. Fats, particularly in the form of fatty acids, provide a dense energy source, yielding more ATP per molecule than carbohydrates. Proteins can also be used for energy, but this typically occurs during prolonged fasting or starvation when carbohydrate and fat stores are depleted. Breakdown of glucose. The main product of glucose breakdown in the mitochondria is acetyl-CoA, which enters the citric acid cycle to produce ATP. This process is crucial for energy production, especially in tissues with high energy demands such as muscle and heart. The conversion of glucose to acetyl-CoA involves several steps, including glycolysis, pyruvate decarboxylation, and the citric acid cycle. Energy utilization by organs. Both muscle and heart tissues can utilize glucose and fatty acids, allowing for flexibility in energy sources based on availability. During periods of fasting or low carbohydrate intake, these organs can switch to fatty acid oxidation to meet energy needs. The brain, while primarily reliant on glucose, can adapt to use keto acids during starvation, showcasing metabolic flexibility. Fatty acid metabolism. Types of fatty acids. Fatty acids are categorized into short-chain, medium-chain, and long-chain fatty acids, each with distinct metabolic pathways. Long-chain fatty acids require carnitine and the carnitine palmitoyltransferase (CPT) system for mitochondrial entry, making their metabolism more complex. Medium-chain fatty acids, MCFAs, can enter mitochondria directly, making them easier to metabolize and a quicker source of energy. Short-chain fatty acids are produced by gut microbiota and play a role in gut health and energy metabolism. Implications of carnitine deficiency. Carnitine deficiency can lead to severe metabolic issues, particularly in preterm infants or those with genetic disorders. Symptoms include muscle weakness, seizures, and fat accumulation due to impaired fatty acid oxidation. This condition highlights the importance of carnitine in energy metabolism, especially in tissues that rely heavily on fatty acids. Ketone bodies and starvation. During starvation, the body depletes glucose stores and begins to break down fats, leading to the production of ketone bodies. Ketosis can provide an alternative energy source for the brain and other tissues, but excessive production can lead to ketoacidosis, a dangerous condition characterized by acidic blood. Understanding the balance of ketone production is crucial for managing metabolic health, especially in diabetic patients. Cellular degradation mechanisms. Role of lysosomes. Lysosomes are cellular organelles that digest unwanted materials using hydrolytic enzymes, playing a key role in cellular homeostasis. Lysosomal storage disorders, such as Gaucher's disease and Tay-Sachs disease, occur when there is a deficiency in these enzymes, leading to harmful substance accumulation. These disorders can have severe consequences, including neurological impairment and organ dysfunction. Proteasome function. Proteasomes are responsible for degrading misfolded or damaged proteins, ensuring protein quality control within the cell. In conditions like cystic fibrosis, defective proteins are targeted for degradation before they can function, leading to disease symptoms. Proteasome inhibitors, such as Velcade, are used in cancer treatment to disrupt the degradation of proteins that promote cell survival, nutritional deficiencies, and metabolic disorders. Consequences of nutritional deficiencies. Severe nutrient deficiencies, as seen in conditions like anorexia nervosa, can lead to muscle wasting and organ failure due to the depletion of energy stores. The body prioritizes essential functions, leading to the breakdown of muscle and fat reserves for energy. Understanding the metabolic consequences of malnutrition is critical for developing effective treatment strategies. Cachexia in cancer patients. Cachexia is a complex syndrome characterized by accelerated degradation of carbohydrates, fats, and proteins, leading to significant weight loss and muscle wasting. Unlike normal starvation, cachexia often results in fat gain without muscle recovery, complicating treatment and recovery. This condition highlights the need for targeted nutritional interventions in cancer care to improve patient outcomes. Testing for fatty acid metabolism disorders. Infants with failure to thrive should be tested for fatty acid metabolism disorders, as early diagnosis can lead to effective dietary management. These disorders can significantly impact growth and development, making timely intervention crucial. Specific dietary adjustments can help manage symptoms and improve metabolic function in affected individuals.